So our next speaker is Dr. Christopher Gobler, who's a professor here at SOMAS. He was also a PI on a project with a special focus area that Sea Grant funded back in 2004 to 2006 on trying to determine what might happen to the biological resources after a breach. Now, at that time, there was no breach, so the idea was how can we potentially simulate the conditions, and they did a gradient study across the length east-west of Great South Bay, and looking at nutrients and phytoplankton, shellfish, and SAV, and so it's really great to have had that baseline now. And Chris is going to tell us a little bit about his results from that study, as well as some of the work he's currently being funded to do by the National Park Service.
Okay, great. And I'll probably talk more about that former project in my second talk that's focusing on the hard clams. And I'll start by acknowledging my co-investigators in the project, Jackie Collier. Jackie, I do have your slides, and I'm going to... How are you feeling about coming up to present those two? Okay. That would be great. And then Darcy Lonsdale is also here, and she's working on the zooplankton parts of the project, which we're actually not going to present much on today. So this is when we had a group here at SOMAS working on all of Great South Bay. This is a nice diagram we had put together to represent the whole food web of Great South Bay by a great local Long Island artist. And well, we're starting to move towards the biology, and for what I'm going to talk about mostly for my talk is the bottom of the food chain. Here's more towards the top, but the phytoplankton, also thinking about things like nutrients and general water quality. So we've been out to the breach about 40 times since it first happened. Ryan Wallace is here. Raise your hand, Ryan. There he is. He was out there in December 2012, and he's been out there organizing the efforts out there since then. And we're going to keep doing this on a weekly to monthly basis for at least for the next 2 years. We have a series of stations that we've been sampling. These are our discrete stations that range from within the Forge River, since we had a lot of good historic data there, and then also matching up from that New York Sea Grant project a few years ago. These stations, this one, that one, and this one, all match up with what we did in 2004 and 5, so we can compare back to that. And we've got a few other stations in there to make sure we have a good gradient across the whole system. And then here is the system that Ryan developed, specifically his data flow system that we are now using on our vessels, whereby the vessel can run at a high speed and continuously collect data with simple parameters: temperature, salinity, oxygen, pH, chlorophyll. We're also using something known as a fluoroprobe to get multiple pigments, and it's all grounded with GIS measurements, so it's georeferenced. And here's the intake for at least showing you the water going into one of those probes and it being spit back out. And this is, I think, from last year, since become more sophisticated with multiple probes in line. And here's a general cruise track that we've been doing. This is what we do in the winter. In the summer, we actually go all the way from Southampton to get data on all the inlets. But we are very interested in the eastern part of Mauritius, the western part of Mauritius for this study as well. And you'll see why that is momentarily. So we set forth a series of hypotheses when we started the project about how the breach would affect water quality and other aspects of the bay. They're all seen here. And we were right on some. We were wrong on others. Let's see what we found. The first was the most obvious, and that has to do with salinity. And we know, of course, that our ocean inlets are important for bringing salt and ocean water into the bays. So this just shows you a representative cruise track. This is from actually summer 2013, one of those cruises where we began in Southampton and went out to Captree and back. So this is that data flow system. And it shows very clearly the fact that near the now 4 inlets we have over this track, when you get near those inlets, you get to that near ocean salinity in the estuaries. And it also shows, importantly, I think now across this track, we have a series of what I'd call nodal points where it's the lowest salinity, and we can infer from there that these are the areas that have the longest residence time and the poorest flushing. Looking a little bit more carefully, here's one of our earliest measurements of salinity, and this is a trend that we've seen going forward. Now, Ryan's put in some kriging methods here to sort of smooth the data out, but note the salinity here is very high, both within Belport Bay, but also going to the east in western Mauritius. And that's a trend that seems to be emerging over and over again. So you can see, obviously, the salinity's gone up. Charlie showed that. But what we're seeing is that ocean water seemingly communicating quite well with the Mauritius Bay Inlet, maybe not so much within Greater Great South Bay. And that's shown again here. Here's a particular cruise after the one we just looked at that shows a similar pattern. In this particular case, I think it was on an outgoing tide. But again, even on those scenarios, you see that ocean water getting through there, but not necessarily seeing that within the bay. And then finally on the salinity front, Ryan put this plot together, and this shows the salinity through November 2013, and again you see the same general pattern, that high salinity showing up by each of the inlets. But here's 2014, and what we're noting is that actually we think that the inlet, well, of course we know the morphology of the inlet is changing. We think the extent to which it's flushing Great South Bay and Mauritius Bay is also changing. So now here's comparing 2013 and 14, and here's the change in salinity over a one-year period. And so what it's showing is actually the salinity lower in Great South Bay and higher to the east. Now part of this may well be that large flush of fresh water that we saw from August. I'm going to talk more about that, flushing through the system. But that wouldn't explain this, right? And so what this, for sure, a higher salinity suggests that now the inlet is flushing more to the east than it is to the west. Moving on to chlorophyll now and phytoplankton blooms and phytoplankton communities in Great South Bay. Here's again a representative plot using some Suffolk County data over a 35-year period and comparing that to what we saw in March in 2013. And again, this pattern continues to emerge where, it's hard to pick out strong chlorophyll differences here, but clearly lower levels now in Belport Bay, Narrow Bay, and also in western Mauritius. And so we might call this the reach of the breach, perhaps. And again, when we do our overall cruises through this whole system, that's a pattern you see at any inlet. So when you get close to the inlet, the chlorophyll levels drop away in the areas, as I mentioned before, with the lower salinity and the higher residence times. That's where you see the highest levels of chlorophyll across the South Shore. I'm going to now just turn to some data from Suffolk County. Of course, we all know that Suffolk County has done an amazing job of monitoring water quality, probably one of the best water quality monitoring programs in the country. Data going back to 1976 with lots of different stations. I just showed some of that. I'm going to show you 4 plots, and when we did this, we've done this in many different ways. We looked at all the data going all the way back to 76. But as I'm going to tell you momentarily, we had a brown tide in 2013, and the data was only available up to 2013. And so the brown tide threw off lots of things. That is, when you get a year of brown tide, there's more nitrogen in the system, there's more chlorophyll in the system, the oxygen levels are lower in the system. So what we did is a simple comparison of that 2013 data to the years prior, the 2 prior brown tides in 2000 and in 2008, which had similar densities and similar patterns. So we make those comparisons. Here's what we see with regards to chlorophyll. Again, correct, now the brown tide's accounted for. The chlorophyll levels, like I showed before, significantly lower in this part of the bay. Actually, maybe a little bit higher within the central part of the bay. With regards to nitrogen, you can see a significant drop in this area, again, where we think the breach is having the largest impact. Also a change here, but much, much smaller. And I note that the levels here, 0.2 milligrams per liter, is substantial. Just for example, the EPA would like to see all estuaries have a total amount of nitrogen of 0.4 or 0.5 milligrams per liter or lower. Well, Belport Bay was significantly above that before the formation of the inlet. And you can see now with that drop, it's actually at or below that level. So a big impact in that general area. And then water clarity is also a big change. And you can see everywhere the water clarity has improved, and a very large improvement here. You can see almost a meter deeper in water clarity, although I'm wondering if this is meters or feet. Maybe it's just feet, actually. It's not going to be meters. That's got to be feet, so excuse me for that. But importantly, one thing, just as an anecdotal note, I've said this before, maybe people heard me say this, the New York State Department of Health says you shouldn't swim in waters that have water clarity less than 4 feet. It's dangerous. You could dive in, you could hit your head. If you went under, someone might not be able to see you. Bellport Bay was below that standard, or it was more turbid before the breach, and now with this change, it's above that standard. So in theory, the water is now swimmable. Bill, you have a question?
Chris, are these numbers the change, or
these are the change. The change between, again, this is looking at 2013 and comparing it to all the observations in 2000 and 2008 to correct for the occurrence of the brown tide. So this is the change. So all of these are improvements in water clarity. And that was the same here. These are all changes. So higher chlorophyll, less chlorophyll. These are all drops in nitrogen, decrease. And this is all an increase in water clarity, feet, not meters. And then with regard to dissolved oxygen, a similar trend with regards to higher levels of oxygen now, at least in 2013, through much of the bay. Maybe far to the west, no change whatsoever. But higher levels of dissolved oxygen. And then, just to point out for temperature, this is again going back to our long cruise track, but another important thing to note. This is an August cruise, and temperature in August is important. We all know the importance of eelgrass. Brad Peterson's here. He can tell us that the thermal, in a normal year, in the South Shore estuaries, the temperatures can get up to above 29 degrees or at 29 degrees Celsius as an extreme. And you can see here there are plenty of places that are above 24. Now, 24 to 25 is stressful for seagrass. That lasts for a long period of time. They can start to die off. It also leads to the reduced filtration rate by bivalves. I'll probably talk about that in my second talk, but high summer temperatures are problematic for many aspects of the food web. So what you see is that these inlets in the summer, when the temperatures are highest, bring in cool water and actually have a protective effect. And again, those areas that had the lower salinity, higher chlorophyll, presumably longer residence time, have higher temperatures as well. But in this area of the breach, you can see the temperature, the moderating effect that the new inlet has on temperature during the summer. Okay, now to turn to brown tide. It dawned on me just yesterday as I put this together. Happy birthday, brown tide. 30 years on Long Island. Who knew that? We just finished our 30th year of having brown tide. And it continues to be a big deal. These are some of the initial response. Most of us are aware, but just as a reminder, we lost a lot of eelgrass following the first brown tide, much of which has not recovered. Our scallop industry collapsed by more than 99% following the brown tide. We know our initial loss of hard clams was due to overharvesting, but we also know that restoration efforts continued since this initial decline have continued to fail because of continued brown tide. And Carl LeBou, who's right here, could tell us how in 2008, on the brown tide there, all of their, I don't know the percentage, 80, 90% of their sea clams perished following the intense brown tide there. So it has a big effect on the biology of our coastal systems. And so, are we getting relief due to the new inlet from all this flushing? Well, we're not in a situation where we're getting no brown tide. This is some aerial photographs of the brown tide that broke out in 2013. Charlie Flagg at the helm of the ship, me taking pictures, but I believe this is Patchogue Bay. This is obviously looking out to the ocean. And so, in July of 2013, we had a significant brown tide. In the beginning of July, break out throughout the bay. These are very high numbers, over 1 million cells per milliliter, something that hadn't been seen since 2008. You can see lower levels here. Just briefly want to point out what led to this brown tide and others like it that we're having. And as I do that, I just want to point out an important climate change issue that I'm not sure that I'm just becoming cognizant of. So, global temperatures have increased about 8 degrees over the last several years, 2 centuries in fact. Global CO2 levels are up about 40%. But here's the change in heavy precipitation for the Northeast, 71%. That's a lot. And that's predicted from climate change, and that prediction, they're just putting this data together. This is from the National Climate Assessment that came out this year. That prediction was true, that was predicted long ago, and is expected to continue in the future. So this is heavy precipitation. That is all the rain happening all at once. Kind of like what happened in August, which I'll talk more about momentarily. I also want to point out that all that post-rain is happening in a scenario whereby we have a lot more nitrogen in our groundwater. Here's the change from 87 to 2005 in our top two aquifers on Long Island. You can see the increase for the glacial and the Magothy, both of which are likely to influence our coastal waters. There are numbers in 2013. The numbers are higher. The rates are about the same. So increases in freshwater flow, and now that fresh water delivering a lot more nitrogen than it had been. And so here's the diagram I put together to talk about that 2013 bloom in Great South Bay. It started a month before the bloom actually initiated with one of those abnormally large rainfall events. Charlie had pointed this out in his talk. More than 8 inches of rain in just a few days, and this is Charlie's data on salinity, a large drop following that, an initial phytoplankton bloom that happened right after that with that big input of nutrients. That bloom died off, and as that bloom died off, we saw the brown tide take off, over 1 million cells per milliliter, and the change in the way the bay looked. This is a picture from early June, and this is just a month later. So, but what is the effect of the inlet? So on that same flight, this is a picture that I took looking out at the new inlet, and you can clearly see the ocean water coming in to dilute the brown tide that was present. And you can imagine that inlet's not there, that water is going to be stagnant. We just saw that the residence time had been, a long time. I don't remember the number. What was it, 30 days? And clearly, on the tide, it's now flushing significantly more. And so we can see that in some of the data. Here's the last brown tide bloom in June of 2008, and here's the numbers, same area with the inlet present. You can see the numbers significantly lower, maybe an order of magnitude lower. And then following that July 2013 brown tide, as it began to decline from millions to hundreds of thousands cells per milliliter, you can see how quickly within the inlets the water cleared up. So rapid flushing leading to the rapid removal of brown tide, and again, this is one of those tracks where we did the whole South Shore. And a similar trend in the fall of 2013 when we saw a brown tide as well. Again, in the middle parts of the bay, the most intense brown tide, significantly less intense by the inlets. And I should point out my student Yunja here has been counting all these brown tide tables for all these years and helping us to document this. So here's her data that she put together for 2013. Here's that brown tide that we saw in the summer develop, and here's the one that developed in the fall. These are the maximum densities that we're seeing in the middle of Great South Bay, and here's what we're seeing in Bellport Bay at our inlet station. Not zero, but significantly lower. Again, flushing out the brown tide on the tide. So now go back to that county data, and compare what we saw for the 2013 bloom compared to 2000-2008. Significantly lower levels of brown tide throughout, so this is all negative, so less in 2013. But the difference being a little bit less so on the eastern side here. Okay, now just to talk about this rainfall event, it was called the 2000 year rainfall event. For those of you who don't know, this was the largest rainfall to ever occur in New York State in a 24-hour period. And you can see this is, Ryan put this plot together using meteorological data. You can see this large swath of rainfall that had over 10 inches that fell quite quickly. And we fortuitously did a cruise before and after that, so here's the interpolation of that cruise, and you can see the fresh water pouring out of the Connecticut River and the salinity drop from in that area of the where the main rainfall fell, the salinity dropping in that particular area. I bring that up here because it was following that rainfall event that I said I think this is going to lead to a brown tide for the same reasons we saw in 2013. Rainfall event, very large rainfall event, very large pulse of nitrogen. We saw a bloom of something else take off before that, Picocyanobacteria. That decayed, and here's the brown tide, and here is the data as of just a few weeks ago. And it's still going on. You can see it hit a peak of almost 900,000 cells per milliliter, currently at about 500,000, and again, The inlet seeing some, but nothing really compared to what we're seeing in the middle part of the bay. So here are the 2 years of brown tide with the inlet. So we had a spring and fall bloom in 2013, no spring bloom in 2014, but again, that bloom coming back in the fall of 2014. And again, I would argue that these two events were stimulated, at least in part, by that cascading effect of the rainfall, pulse of nitrogen, and then another bloom, then followed by a brown tide. And here's some nice aerial photographs from the U.S. Coast Guard, Auxiliary Coast Guard, during the fall bloom. And here you can see, I call this the New Inlet spits out brown tide, but you can see this massive plume of brown tide coming right out of the inlet here. And again, if that inlet's not there, that's just staying right there in Belport Bay. And a few more pictures, because I think they look pretty cool. Here's the inlet once again, the brown tide going out, the clean ocean water coming in. Similar scenario, just a different perspective. And then just very briefly on, with regards to brown tide, here's that 30-year data set. So we've had 3 out of 4 seasons brown tide with the inlet in place. It's too early to say what that means. But initially I would have guessed, or I would have predicted that we're not going to get brown tides with the new inlet, and I was wrong. And instead, like I said, out of potential 4 times we could have had it happen 3 times. And you have to go back kind of far to see that in the long-term data set. So the brown tide is not being fended off entirely within the central part of the Great South Bay by this new inlet. And I just also, for fun, since it's 30 years, here's the first 15 years, the next 15 years of brown tide, comparing spring and fall, just to simply note whether a bloom occurred, yes or no. More frequently in that first 15 years in the spring than fall, and now we're seeing almost the reverse, as we're seeing more frequent fall blooms of brown tide than in the spring. That might be a climatic effect. If you look at the seasons, fall temperatures seem to be warmer further into the season, maybe allowing a longer window for brown tide, which does not like those high summer temperatures. Okay, Jackie, do you want to come in and say a few words? Jackie's using, well, she'll tell you, she's using her flow cam to, and maybe you'll want to be by the microphone. Since
Okay, I'll try not to drop it. So, a little over a year ago, we decided to use an instrument we've had around for a while called the flow cam. It's kind of a cross between a flow cytometer and a microscope. Basically, a flow-through microscope that captures digital images of every particle it detects that, meet certain criteria you define. So it doesn't look only at phytoplankton, it also will capture zooplankton, and much to our surprise, it's revealing how many particles in the water are detritus in Great South Bay. The data I'm showing here go from, this is dangerous if I push this button, right? From, apparently.
It's a little red one in the middle. The little red guy.
That one. So we go from September 2013. This data only goes through June. We decided to change how we were analyzing some things, and we don't have everything together yet. So these are only for particles larger than 25 microns. And all I want to really show is that this is the Fire Island Inlet Station. These are the 2 middle of Great South Bay stations Chris was showing, and this is the station nearest the Sandy Inlet. And I just want to say that during that first fall and winter, the 2 inlet stations, in terms of the large phytoplankton and in terms of its general distribution of particles, looked much more similar to each other and very different from the stations in the middle. And they shared a large amount of very big diatoms, things like Eucampia, Titoceras, Rhizoselenia, that just were much less abundant in the middle of the bay. This top panel is in terms of our estimates of the total biomass of carbon per liter in micrograms per liter. The bottom is the same data just presented as percent of the total biomass. That difference between the inlet stations and the middle Great South Bay stations disappeared by about late March, going into April this past spring. And through the spring and summer, there was no longer a dramatic difference between the 2 inlet stations and the middle of Great South Bay stations. And this surprised me. I would have expected to see a bigger difference through the summer, then during the fall, and I don't have an explanation. Thought I would point it out. How are you advancing it, Chris?
Yep.
Okay. This is just a clustering analysis that basically shows the same thing. There are, if you separate the branches here and here, there's two main clusters of samples. The top cluster includes all the inlet samples except for one, and it includes all the May and June mid-Great South Bay samples. The bottom cluster is all the mid-Great South Bay samples from September through March, and then this one inlet station sample. So, again, in the fall and winter, there was a very distinct difference in the plankton communities overall. This is for all the data, not just the greater than 25 microns. But during the spring and summer, something changed, and they are no longer as different as they were.
Well, as you mentioned that, the one thing I will say is we do know that in the middle of the bay there was less chlorophyll. Bob Serrato's here somewhere. He might tell us about. He'll be back. The mussels filter in the water. We know the water was a lot clearer in 2014 until the onset of the brown tide and a large rainfall event. So that may be why, keeping the check on the phytoplankton. Okay, and I think I just have one last data set to show that I think is of interest. It's about a dinoflagellate, probably one people don't typically think about on Long Island, but it is a toxic dinoflagellate. This is Dinophysis acuminata. It makes this very large toxin known as okadaic acid that causes diuretic shellfish poisoning. And we've had this on Long Island for a long time. There's a great paper that was published by the Nassau County Department of Health that actually looked a little bit at Suffolk County as well and showed this was widespread on Long Island in the 70s and the 80s as well at low levels. But we're now seeing significantly higher levels today than they had seen in that prior study. So this is densities we've seen between 2011 and 2013 of dinophysis. You can see only one location where we didn't find any cells, and I think we found them in 2014, so that's going to be a certain color. But you can see it's very widespread, and in some areas we have problems with it, hitting very high densities, over 100,000 cells per liter, notably here in the Flanders Bay region, Meeting House Creek, but also in the area of Northport and Huntington Harbor. And again, not only the cells there, not only do the cells make toxins, but the toxins are getting into the shellfish. The U.S. FDA says that shellfish should not have more than 100 nanograms per gram of okadaic acid or dinophysis toxins. Here's some surveying that Teresa Hattenrath did along with the DEC in 2011. And you can see for multiple locations, the shellfish were above that level. Now, thankfully, this is an area that's closed to shellfishing, because we're seeing levels that are almost an order of magnitude greater than the FDA limit, although this area was above that and above the FDA limit, and this is an area that was open. So this is an emerging issue, I think, in New York State. We happen to be looking for this in Bellport Bay, both in 2012 and 2013, and I have that data here. So you can see we had and before the inlet being present, we had the emergence of dinophysis. Not so ridiculously high levels like we're seeing in Northport, but 1,000 cells per milliliter. And you can see in 2013 that was not the case. So we need to look at some further data, 2014 prior data as well. But at least with regard to this toxic dinoflagellate, at least in Bellport, it looks like it's at very low levels with the presence of the inlet. So overall, our hypotheses, many of which were on the money. We do see less brown tide near the inlet, but we're seeing potentially more frequent brown tide. It's too early to actually say that. That's why there's a question mark. But it's 3 of the last 4 potential seasons it's been there. There's less toxic dinophysis, and all the data points to strong tidal flushing to the east, and the middle of Great South Bay still being somewhat sluggish compared to not flushing as well. I'll just leave it at that. So I think that's my last slide. Bill.
Chris, what's the recent history of Burgess Inlet, Ocean Sandy?
Well, I think, Jay, was Moriches Inlet also dredged? I think it was.
Yeah,
I heard a yes.
I don't know exactly where it was dredged and how that would affect the flux of water through it. They did take some out of the deposition basin outside of the ocean. And we did see pictures, at least, of the dredge there, but we haven't been able to find the surveys.
Yeah, I mean, one thing I'm just going to point to on this map right here. When we started working in the western bays, Somas looking at the western bays here, here's the Jones Inlet. And when I saw that inlet, my assumption going into the project is that, the ocean water comes in and flushes. And what we actually found out, that's not the case at all. And in fact, all of this water here looks like ocean water. Ocean-like salinity is quite low here. And so it looks like these two inlets are flushing quite well. And so potentially now, at least what it looks like is that this new inlet is flushing quite well with the Moriches Inlet, leaving this area, sort of, well, I don't know.
We need to cut some more inlets.
There are people. Marshall Brown of the Great South Bay says we need 10 new inlets in Great South Bay. So. Yes, so with support from the National Park Service, we're looking at Suffolk County and the New York State DEC's record of indicator bacteria, total coliform, fecal coliform. This is an important issue for Great South Bay. Great South Bay lost, maybe Carl can remember the precise number, I think at least 2,000 acres of shellfish beds because of higher levels of coliform bacteria coming up in the central part of Great South Bay. It's also an important issue because following Sandy, what we saw, and the DEC data showed, is that the levels of coliform bacteria jumped way up following Sandy and stayed up surprisingly until January. So we had a 2-month period of levels of coliform bacteria that were above permissible for harvesting shellfish. So clearly, that's an important issue. So we've compiled that data. So far, what we've really focused on so far is that response to Sandy, because we were very interested. We know that DEC had closed the shellfish beds. We hadn't looked at the data. So far, the data shows this very quick spike following Sandy, and again, leading on into 2013. We're now honing in on the area of Eastern Moriches and Belfort Bay to get a better sense of the dynamics there. But we really just started that project, so no data on that aspect to present right now. Well, we're relying on the DEC's data and Suffolk County's data. We're doing that analysis now. I believe next summer we may do some targeted sampling to supplement that as well.
Chris, your analysis of the Suffolk County Health Department's data for 2013 and then for the pre-breach brown tide years of 2008 and 2000 showed a decrease in total nitrogen. Do you recall if that was primarily inorganic nitrogen or organic nitrogen?
That was total nitrogen. I don't know. I'm trying to remember. If my student Jake was here, he could help us on that. I think it was mainly probably the dissolved organic pool, but I have that data. We have plots for that. We've broken out the ammonia. I think there was a signal in the ammonium as well.
I mean, it'd be interesting to know if it was more a change in primary productivity, recycling, versus dilution of nitrogen coming from groundwater.
Yeah. I feel like the day. I think it's. I remember ammonium is lower, and nitrate was actually a little bit higher afterwards. But anyway, I have that data. I can be happy to share that with you. Sorry that I can't recall it right now. Charlie.
Chris, what's the more important factor to triggering a brown tide? Is it the reduction in salinity or the increase in nutrients?
Well, they come together, obviously, and its niche is high levels of organic nitrogen and low levels of nitrate. And therefore that's why following the large pulse of fresh water, you don't see it at all. Something else comes in. So of those two, I'm not sure which is more important, but what the mechanism is, remember, is the fresh water delivering the nitrogen, stimulating something else that then dies off and liberates that organic nitrogen that the brown tide feeds off of. And then, of course, because it's it's not palatable to both shellfish and zooplankton. Not a lot of things eat it, and it sort of gains a foothold. I don't know if that answers your question.
Any other?
Chris.
We're going to go back further historically and look at that correlation of rain events to blooms.
We have seen that actually historically. For example, I was studying brown tide a long time, I guess, but during my doctoral dissertation, we saw the same pattern in the Peconics, where a pulse of rain, something else takes off, it dies, and then the brown tide takes off. So it is there. It's there in the historical records. I don't know if it's a prerequisite for a brown tide. We've probably had the initiation, at least in the spring, without that sort of event, but it certainly, I think, helps kicks things off. Yeah?
I have a biology 101 question. What is brown tide?
Sorry about that, yeah. Brown tide is a phytoplankton. It's specifically a pelagophyte, and it's about 2 microns in diameter, very small. And so it's, as a phytoplankton, the base of the food web. And when it grows very dense, because it can get to about 1 billion cells per liter, it starts having these nefarious effects across the ecosystem, excluding the good types of phytoplankton, and then as the only phytoplankton or eukaryote out there often, it's not a good food source for many things in the food web, zooplankton, shellfish, etc. Sorry.
Thank you.
You're welcome. Bob?
And what makes it go away? What makes a bloom?
Well, in the summer, that's an easy question, because in the summer, the temperatures almost invariably go above 24 or 25 degrees, and it can't hack that. It's got a temperature window, and the upper threshold is around 24. So that makes it go away. In the fall, it's anybody's guess. In 1999, the brown tide that initiated in the fall of 99 persisted Suffolk County. Mike, were you there sampling under the ice? Somebody was there collecting samples, according to Bob Newsy, under the ice, and it was still there. So in the fall, it's not as clear because it can handle those lower temperatures. I expected it to go away more quickly this fall, temperatures going down and enhanced flushing, but it seems to be, well, we'll see. We have the next cruise Monday. We'll see where it is at that point.
All right. Well, thank you, Chris.